Hi, my name is Kevin Lester from Hayti High School. I am a junior, and my project was the crystal structure of 2-amino 4,6-dihydroxypyrimidine determined from X-ray crystallography. Okay, and X-ray crystallography is a science that helps us determine the structure of crystals, and after the process, you can determine the atoms' locations and the bonds that helped it form. And we chose this process because it is important because the results are accurate enough for it to be used in forming many pharmaceuticals today. And this is the picture. Okay, and the problem of our experiment was to see if we could produce a crystalline structure using a pyrimidine powder. And to do this, to get many possible outcomes, we mixed a lot of various mixtures to see if they will result in crystals. Okay, and after we made the mixtures and left them to grow, 6 of our mixtures had produced crystals. And after this, we took the crystals out of the test tubes, which we had put the mixtures in, and transferred them to the Bruker analytical X-ray system, which underwent the process of X-ray crystallography with the crystals. And from the results of the process, our first mixture contained 2-diamino 6-hydroxypyrimidine, adenine, and nitric acid, and it was identified to be adenine from the results. And this is a picture of it. Wait, it skipped. Okay, and our second mixture showed results. Okay. From the results of our second mixture, we had got 2-amino 4,6-dihydroxypyrimidine. And this is a picture of the crystal and a picture of the crystal undergoing the process. And this is a structure of our crystal that has been shown after the process of X-ray crystallography has been done on our computer. And then on the left and on the right is a handwritten model. And this is an example of the process that we think happened to form our structure. Okay, and for my conclusion, oh my gosh, okay, let me slow down a little bit. Okay, conclusively, our experiment is not yet finished, but this is what we have found so far. We have yielded 2-amino 4,6-dihydroxypyrimidine, which has no evidence so far, so it may be brand new, because there's no supported evidence of it existing. But we have other crystals that have yet to be tested, which we have not gotten to yet because we have not got back to our laboratory. And applications of this discovery are basically not unknown yet since there's no evidence of it, but it is a nucleic acid and it can have many applications in pharmaceuticals, research, and in pharmaceuticals, and cancer study, and epigenetics, and can be used in research for DNA repairs. And this is the structure of the adenine crystal, which formed from our first mixture. And this is the 2-amino 4,6-dihydroxypyrimidine and its structure. Okay, and I would like to give a special thanks to Dr. Marcus Bond because he was our guide through the experiment. He gave us a lot of information as well as provided our materials. I would like to thank you for having me. Any questions before you continue? I apologize.
You did excellent. So Kevin, one of the things that I find the most amazing about your project is the fact that what you guys have actually done is created a crystal that allows for, which is a nucleic acid. And so it allows to actually be used in a pharmaceutical product. Yes. And because this is really the first time that there's been a pyrimidine that would actually attach to the DNA in a very specific location, which from a pharmaceutical standpoint, what this would mean is that, the way I understand it, and you can correct me if I'm wrong, is that this would allow for the first time specific genes to have their expression suppressed, which would mean stop a disease.
Yes, the polarity of our structure means that when it connects to others of its kind, it forms B sheets. And when it attaches, it could block particular like you said, particular ones from passing through, which could in turn help prohibit diseases and genetic diseases.
So we have a question. It says, I actually looked at your project at the science fair. I know you had the issue with the hydrolysis of your amino group. I proposed a mechanism for this hydrolysis, and the presence of hydrobromic acid seems to be catalyzing the hydrolysis. You should be able to prevent this hydrolysis in the future by using a solution of hydrobromic acid and alcohol. Have you considered doing this?
Well, we were going to continue our experiment by running hydrobromic gas through it, but we did not get to do that before the shutdowns happened. So there are a lot of things we would like to continue our experiment with doing to see what happened.
And the mechanism involving acid catalyzed tautomerization, I'm not sure I said that correctly because I'm not actually sure what that means, followed by a hydrolysis with excess water molecules. I hope you know what that means because I sure don't.
Yeah.
All right. Well, since you're in 11th grade now, is this something that you plan on doing next year? Continuing on with the research?
Yeah, I plan on continuing with this experiment to see what exactly caused the process to undergo to form the structure. Because they like I said, this is the first time anyone has gotten this structure from a pyrimidine. So I would like to see how it's formed because we have one and nobody's ever got one from it, from a pyrimidine.
So this could be really foundationally changing the way that pharmaceuticals and or expression of diseases that are based in genes. So yes. Congratulations to you and your group, and we look forward to seeing what you have next year.